Good afternoon, everyone. Just out of my curiosity, could you tell me what your background is? What do you guys study? Chemistry, biology, mathematics? So biochemistry? Yeah, biomedical. Cool. Oh, right. So that's you already have quite higher up on your doing a PhD or? Oh, you're all PhD students. All right. Well, I will talk a little bit today about what we are doing in our lab in Nottingham. This is one of many things we do. I must admit, it's not the only one. And this is all about in silico studies on G protein coupled receptors and how can we design new ligands for them, hopefully one day. So maybe if you don't know where we are based in Nottingham, so we are center of membrane protein and receptors. And this is about 30 PIs joined between the University of Nottingham and the University of Birmingham. And we do have one group at Coventry who's joined us. And we're really focusing on G protein coupled receptors, receptor tyrosine kinases. So the membrane receptors which decorate the surface of cells and mediate signaling responses, over hormonal responses which are being transmitted through our body. And we have quite a large variety of skills in our groups of 30 PIs, ranging from biochemistry and biophysics and structural biology all the way to animal work. And somewhere in between, there is a classical pharmacology of radioligand binding assays and signaling assays. I'm more on the biophysical and structural biology side of things. So what are G protein coupled receptors, or GPCRs, as we like to call them? So this is, these membranes, these proteins are sitting in a membrane, and all of them share the same three-dimensional architecture. So there is 7 transmembrane helices, which are arranged in a particular way, and the ligand binding site for hormones somewhere in between these helices. And on the inside of a cell, they can recruit G proteins, and they can recruit our class of proteins called G protein coupled receptor kinases, which phosphorylate them when the receptors are activated by hormones or by drugs. And of course, when the signaling is being stopped by proteins called arrestins, which mediate their own signaling pathway through MAPK pathway. So that's a very kind of a simplistic view of what GPCR signaling looks like. But what you have to remember that, we have a lot of GPCRs. As it turns out, this 7TM motif is extremely useful in nature to recognize molecules. So it has been reused over 800 times in humans. So we have approximately 350 hormonal receptors, adrenaline, chemokines, you name it, all sorts of different hormones have been recognized by GPCRs. And all of most olfactory receptors are also GPCRs. So whenever you taste beta taste or something else, or it smells really nice, or it smells not really nice, there are all GPCRs involved. You see light, this is rhodopsin in our eyes, this is also a GPCR. So we have, as I said, that's kind of a tree of life of GPCRs. And today we will focus a little bit on 2 receptors here, the adrenergic receptors which sense adrenaline in our body. And specifically beta-1, which is in our heart mostly, and beta-2, which is mostly in our lungs. So why GPCRs are so hot? Well, it turns out they're actually very useful drug targets. And the reason is they regulate a lot of different processes in our body, and there is one GPCR for everything, for every process individual. So you can very specifically and selectively target them with drugs. And if you look at statistics, how many drugs are in use today? I mean, I have actually an answer on the slide. But if I did, it's about 1,500 drugs are approved by FDA today. And the rest of the world would be somewhat similar. Of which about a third, so nearly 500, actually working on GPCRs. Things like cannabis also works on GPCRs, on cannabinoid receptors. This is probably one of the first drugs in use in the history of humanity, We have archaeological evidence suggesting people have been using cannabis as medicine 9,000 years ago. From a point of view of genes, which could be targeted for therapeutics, GPCRs are a bit smaller, right? So whereas if you think about it, what kind of classes of drugs are out there, apart from GPCRs, we have ion channels. So we have ligand-activated ion channels and voltage-gated ion channels. We have transporters, which move molecules across the membranes. We have kinases, which phosphorylate things. And kinases are favorite targets in cancers, for example. So we have many, many classes of drug targets, which we target today, and many more to target. So out of this, as I said, there are 800 GPCRs in humans. Approximately 350 are involved in hormonal regulations, and about 400 are in olfactory. And there are some other very interesting classes of GPCRs which are responsible for development and connecting tissues, cells in tissues together, called adhesion GPCRs. Only about 100 is targeted at the moment. So we actually have a huge scope of developing new drugs for novel diseases, or maybe for existing diseases, but new drugs, looking at GPCRs. When you talk about ligands for GPCRs, that's something which comes from a very classical pharmacology, and that's something, so me personally, as a structural biologist, so when I became a professor of pharmacology, I also had to learn it, right? So classical pharmacology, so we have ligands that activate GPCRs, they're called agonists. And you may have heard about it, but I studied different things, so I never really paid attention to pharmacology in my life. And we have ligands which deactivate GPCRs, they're called inverse agonists, or antagonists is another name for it. And the reason is the receptors have some level of basal activity. In the absence of ligands, they would signal a little bit. And this a little bit could be as little as 0 for rhodopsin. It signals spontaneously maybe once every 10 years. And maybe as much as 50% for cannabinoid receptors, which signal a lot in the absence of any cannabinoids. So we have these variations in activity of receptors, but the most important thing to remember from this slide is we have agonists, which activate the receptors, and we have antagonists, which inhibit, or inverse agonists, which inhibit the receptor. And when we want to design a new drug, we have to know what we want. Which one do we want? Do we want to activate our receptors, or do we want to deactivate our receptors? And both are useful in different diseases. So this is perhaps some of the concept of receptor has been proposed over 100 years ago, before we knew what proteins are. Before we even suspected that proteins are, we have a variety of proteins in our body, the theoretical concept of a receptor and of hormone, of a signaling molecule, have existed already. We just didn't believe that there is a physical thing which actually connects them. It was just a concept. But they were cloned eventually in the 80s, so quite a long while down the line. And then people realized, oh, there is actually a whole family of these receptors, because when you start cloning, you can clone things which look similar. They don't have to be exactly the same. And then we think, oh, that looks like rhodopsin. And rhodopsin was a very fantastic protein to work on because it can purify it from native sources, from ice. And when I was a student, we used to go to the butcher and get a bucket of ice and purify rhodopsin from them. So beta-1 adrenergic receptor senses adrenaline. So when you're frightened, what happens first? Have adrenaline release from your brain into your bloodstream. Your heart starts pumping faster. You're prepared to fight or to run away, right? So that's the important aspect of it. Of course, in different tissues it does different things, but that's a job of adrenaline. Unfortunately, when you have problems with your heart later on in life, this is bad for you, right? If your heart starts pumping, you can die. So beta blockers is the best-selling drug in the history of modern medicine. It was developed in the UK, and Sir James Black got a Nobel Prize for this. And that was the first GPCR drug developed in the 60s. So the first beta blocker, beta propranolol, It is not in clinical use today, but it's still used to calm your nerves if you're a sharpshooter or if you're an actor. On the other hand, so beta-2 adrenergic receptor is mostly in our lungs. And if you ever seen people with inhalers, so that's what we have. We have agonists for beta-2 adrenergic receptor, because what it does is it induces opening up the airways in our lungs if you have an asthma kind of attack. So these are very important and useful drugs. We all, well, not all of us, but many of us use on a fairly daily basis. So if not you, then your grandparents probably are on beta blockers, and half of your friends are probably on inhalers. So what does it look like from a structural perspective, right? So you have your 7TM bundle of helices in the membrane, and they are organized in a very particular way. And it is the same for all members of the GPCR family. But of course, there is a devil in the detail. So the sequence identity between these GPCRs, even though in a three-dimensional structure, they look almost the same, is actually surprisingly low. I think on average it's like 10%. You wouldn't, for the rest of the proteins, you wouldn't call it similar proteins even. But in the GPCR world, 25% identity is huge. And 40% identity, some of the boundary for soluble proteins, is like a close cousin. And the reason is this individual amino acids which line up the ligand binding pocket, they actually determine the specificity towards your hormones. So we have a lot of structures. So that's something which was a massive undertaking. To get the first structure of GPCR took forever. So bacteria have a very similar protein called bacterial rhodopsin, has the same architecture, and the structure that was solved in 1986, and it's deservedly got a Nobel Prize for it. So the structure of human rhodopsin, or bovine rhodopsin, because bovine eyes are easier to get than human eyes, was solved in 2000. And after that, the next structure was solved, the adrenergic receptor, in 2007, and it took 17 years of continuous efforts to get there, and solving a lot of technical challenges in the process. From 2007 till today, and that is roughly where I started moving into this field, we now have 600 structures in the GPCR, of GPCRs in different states. And most exciting, but latest with the development of cryo-EM, and one of the reasons I'm here today, actually, apart from giving a talk, I talked to the cryo-EM facility in University of Warwick to start a joint project on determining cryo-EM structure of our receptor. But now we can solve structures of GPCRs complex with G proteins. And the reason is that GPCRs are very mobile, very dynamic proteins, and they look somewhat different in different states. If it's active or inactive, they look a little bit different, and they also bind ligand differently. The ligands bind differently. But we thought, well, if we start zooming into this using computational methods, what can we learn? Right? Well, we have, what is the difference between ligands that activate the receptor and deactivate the receptor? Let's make it very simple question. So here we show the example of two endogenous agonists of beta-adrenergic receptors, adrenaline and noradrenaline, and propranolol, which is it's an inverse agonist or antagonist of the receptor. They look somewhat similar, but not exactly the same. So there are little minor differences. Here, you have this extra methyl group. Here, we're losing hydroxyls, but we replace them with something else. So how does this make them agonists or antagonists? And now I'm coming to a slightly different topic, machine learning. And the reason is that we have to use machine learning to answer these questions. And machine learning, you probably all fit the data in Prism, right? You take your data points, you fit them to a curve. And that is an example of rather simplistic machine learning. It could be one-dimensional, two-dimensional. And in general, machine learning is nothing more than data fitting. it's a far more complex data fitting. You may have thousands of variables to fit simultaneously. And in general, we separate the machine learning in the two kind of techniques. One is, yes? Is a question or? We separate the techniques in so-called supervised machine learning. We're trying to learn something about the process, the inputs, the correlation between the inputs and the outputs. And that is, for example, we have our ligands, and we know whenever they are agonists or antagonists. And we try to learn something which links their properties within answer and make a prediction. And the other question is, we say we have a bunch of objects, like a pile of objects, and we try to classify them by some kind of principles, and 2 piles or many piles. And that's called unsupervised learning. So just try to separate, So we have a lot of chairs of different colors and different shades. And most of you would have little problem separating them in bluish and yellowish. For example, if you want to separate them in 2 piles. Or maybe you want to separate them in 4 or 5 piles, right? Or maybe there is some secret meaning to this. There is one chair. If you sit in it, you get in the top grade. Always. But you have to know that. And maybe a very simplistic example. Let's take a look at the money. Right? That's a very simple object. We all can separate notes from coins. That easy. Right? One is round, one is something, one is made from metal, one is made from paper. That's your easy separation. But now suddenly you have pounds and dollars. And now the problem becomes a bit more difficult, because now you have to know more about coins to say, okay, this is a cent and this is a penny. And the same for the paper notes, whatever it is, $1 or £1. So you can describe your objects and give them properties or features. And that could be anything. That could be weight, could be value, could be color, could be material, could be anything. And in our types of classification of ligands, we actually have approximately 10,000 different properties assigned to a ligand. And then, of course, we have to know the truth. Is it an agonist or antagonist, or is it a pound or a coin? And then you have to make a prediction how likely it is, because the truth, I put it in commas, because in real science, the truth is never 100% truth. So there is noise in the data. Even though you think you know the answer, you've measured it, in large data sets, you'll be lucky if you're 90% correct. And that's important. So what kind of questions are you going to answer? Some questions are simple and easy to answer, and some questions are far more trickier. Because what if you have fake money in this pile? Now, can you distinguish them by combining the descriptors of the objects? So that is kind of a... So one of the very simplistic things which we all do is called decision tree. Go right, go left. Is the paper, is it metal? That's a very simplistic decision tree process. But then, of course, you start questioning, okay, what if I have to have a more complex answer? Then you have multi-layered decision tree. Then they're called decision tree because they look like trees. They always have branches, right, left, decision points. Okay, say, okay, if it's a metal, right, that must be a coin. If it's not a metal, could be plastic, could be paper. If it's paper, that's old pound note, it's no good anymore. If it's plastic, it's a good paper note. Okay, wonderful. But decision trees are simplistic schemes, right? And if you try to answer very complex, make a complex prediction, a complex classification job, you will make mistakes. So the people come up with the idea that they can create forests of these trees, and they're called random forest. So you can choose any split point, any feature could become a split point, and you can just randomly pick them, and then have 1,000 of these trees, and then you train them to say, okay, these trees predict better than others, and then you look at consensus. What do they predict on average? Maybe 70% of trees would say it's a coin, and 30% of trees would say it's a paper. Then the prediction, the overall prediction, would be that's whatever it is statistically more probable. So these are called random forest classifiers, and there are better and better modifications of this are more computation intensive, less noise sensitive. So that's kind of one flavor of machine learning people often use to predict stop oxidase change or ligand binding. So the only problem with machine learning data that you need a lot of data. So this is really a problem. And when we say, oh, we want to look at the structural basis for drug action on GPCRs, you know, you need a lot of data, but we don't have a lot of structures. What can you do? So the best way to generate a lot of data, if you don't have them, is to actually generate them. So you need some kind of a generator of probable, of true-like data. And in drug design, we have in silico docking algorithms which can do that with a certain degree of accuracy. So you know what your ligands look like. You can take a look at the library of ligands. And of course, if you think about every possible ligand under the sun to the size of 500 Dalton, that's a typical drug is about 500 Dalton. That makes it the size of about 40 atoms. And that means that you can actually create an endless number of ligands, so larger than the number of atoms of the universe, just through sheer combinatorics. So now suddenly that becomes interesting because now we really cannot enumerate, we cannot look at every example of a drug. So we have to learn some kind of basic principles behind it to design new ligands. So that's the trick, right? So if you can just list, I don't know, if you have 200 objects, you can actually look at all 200 objects. There's nothing much more to learn there because you can do it. If you have 200,000 and you have half an hour, then it's difficult, right? You have to know the basic principles to separate them into the piles or predict them if they're going to be agonists or antagonists. So we went through all the public databases. People have been designing ligands for these receptors forever. Many thousands of PhD students have designed a ligand on two for them. So there are 2,500 ligands in the public database, of which about for half we know what their pharmacological activity is. So we computationally dock them. So that took a while, but you go through all the databases and curate them, and then you have, okay, I have a decent data set. So then you computationally dock them, and the docking processes today are quite fast. So it takes a couple of weeks on a laptop and a lot faster if you have a faster computer. Of course, it's a very imprecise process. That's a thing. Because what you do, you generate so we use so-called semi-rigid docking. So if you think about how proteins look like and how ligands look like, right? Ligands, I showed you nice two-dimensional pictures of what the ligands look like. Of course, that's not what they really like in real life, right? First of all, they're three-dimensional, and they're very flexible because we have a lot of bonds along which we can rotate. So actually, each ligand may have hundreds of conformations present in solution, and you don't know which one binds your receptor. The receptor is also not rigid, right? The receptor is not a house of bricks. The receptor is actually quite dynamic protein molecule. But because the number of atoms is much larger in receptors, you cannot really create a library of all the possible kind of variation of your receptor. So that's why we call semi-rigid docking when the receptor is rigid, because it's computationally nearly impossible to sample all the conformational space of a receptor. But the ligands are flexible because they're much smaller, and actually it's more feasible to choose 10 or 20 or 100 or 1,000 flavors of the same ligand and then dock it. And then the first step is geometrical docking. It's like a three-dimensional Tetris. Do they fit or do they not? And the second step is try to evaluate if the fit makes sense. Whenever we have steric clashes, or whenever we have charge interactions, or whatever we have certain types of chemical bonds, or certain types of repulsion forces. So and there are many flavors of how you calculate that. But it's all very it's not a very precise science. It's approximate. That means that you get answers that are not 100% accurate. But what it does, it generates a lot of data. And then for us, we have to describe then this in silico generated models in terms the machine learning algorithms can understand them, so they're called features. Right, and each feature in our case is its atom-atom interaction. If an atom of a ligand interacts with an atom of a receptor of a protein, that's a feature. And we can describe what type of interaction it is. Is it charged, ionic bond? What nature of what atoms are these? Are they carbons? Are they nitrogens? Are they fluorides? And so that's a feature. We have approximately each ligand binding pose would create maybe 200 interactions with the ligands, but we all create some different flavors of them. So we have 10,000 different types of interactions. So, and I did mention about the problem of the noise, right? So this noise have a, this data set is probably mostly noise, to be honest. When we manually inspect the binding poses, most of them makes no sense. So I estimate that approximately 90% of it is really random. And that is what I tried to show here, that this is our receptor, and this red bar is a ligand. And when it's dark red, this is its real binding pose, and it makes override interactions with the receptor. But it could also be docked in the different conformations, and then that will be pretty random. So the beauty of it, so if you know something about how the ligand should recognize your receptor, which we luckily do, then we'll create a very filter. You say, okay, the ligand must interact with this particular amino acid. At least then it improves the chances that it's an accurate binding pose. But if you don't have this information, then you have to, you have a full noisy data set. Is it clear so far? Or? So that is what we did. And then we generated this atomic interaction fingerprint, which is 10,000. It's a big table with 10,000 columns. And of course, there are 70,000 rows in it because we generate 70,000 poses, 10 for each ligand approximately. And it's mostly populated with zeros because in most cases, each ligand only has 200 interactions out of 10,000. And that allows us then to start calculating the interactions. And if you think about, very simplistic, you all heard about Pearson correlation coefficients. You take two columns and you say, how correlated are they? And it basically just says, okay, if there is one in column 1 and there is one in column B, and that happens in 50% of the cells or 70% of the cells, there is certain correlation between the columns. And if it's completely random, correlation will be 0. That's a very simplistic way of looking at it, but actually works. Or you start using machine learning methods, which are much more advanced and are much more resilient towards noise. And I think that's what you have to use sometimes. But it's good to kind of have a basic understanding what your data look like. And we start picking up with very strong correlations, which are responsible for agonism, and yellow ones are responsible for antagonism. So now suddenly your ligand binds the receptor, but it turns out the interactions it makes to the receptor actually is very selective. So first of all, not all of them matter. You have 30 amino acids in the binding pocket, and 10 of them, only 10 of them actually mediate any sort of action. And some of them specifically mediate agonism, and some of them very specifically mediate antagonism. And if we use machine learning, we start getting even more kind of a fine-grained model of this. This is called structure-activity relationship in medicinal chemistry. But this level of detail is something you've never heard of before. Because structure-activity relationships have always been very simplistic. Oh, I put a methyl group here, I modify my ligand, and I have an improvement in a binding or decrease in binding. Or I have more agonism or less agonism. But here we have very, very specific. If I have an oxygen here, it's an agonist, and a nitrogen, it's an antagonist. If it interacts with this particular atom of a side chain, it's an agonist. Otherwise, it's an antagonist. So you start seeing a lot more, and you can really fine-tune the action of little modifications of a ligand can have dramatic impact on the properties of whether it's agonist or antagonist. So that becomes really exciting. And machine learning has been treated like a black box for many years. So well, machine learning learns something, but I never tell you what it learned. So luckily, there is a whole branch of machine learning called interpretable machine learning. And in the last 5 years, approximately, things changed. We can actually interrogate your models and have even universal framework, which allow you to interrogate your machine learning model to figure out what the hell did it learn. And that takes 10 times longer than actually generating the model, because you have to make a lot of predictions and do a bit of a, but nevertheless, you can do it. And this is called Shapley value analysis. And what's really exciting, for example, this is the most predictive interaction in the whole data set. And this particular one. No, that one at the bottom. And we knew it already for years, but this is a really important interaction for agonism. So it's not like we discovered, in this particular case, it's one of the reasons we picked beta-adrenergic receptors, because it is perhaps the best studied example. So when you develop a new method, you actually want to develop it into something where you already know what the answer looks like, to know if you're getting the right answers with your new technique. But here we see that the presence of this interaction actually mediates agonism, and the absence of it doesn't really do much. Though in some other cases, the presence of interaction mediates antagonism, and the absence of it doesn't really do much. So now you start looking even more detailed. You know, do you have to have this interaction, or does it really matter if you don't have it? So that's really exciting. The question is, can we do predictions? You know, on one hand, what we have done, we've learned what's important. The question is, you know, for any new ligands. So it's like rationalizing things in the past and predicting things in the future, right? So retrospectively, 1 month down the line, the mini budget was an obvious disaster, right? A kindergarten child can predict that. 2 months down the line, it was not that clear to anyone, right? So predictions is actually quite difficult. So, and the reason is that our docking poses are not accurate. So I can figure out, because the wrong docking poses are filtered out when you average using machine learning or correlation coefficients. So you basically, random noise gets filtered out from large data sets, and you only get the really important interactions. But when you make the predictions, you don't have a luxury of 100,000 binding poses anymore. You have only one. And suddenly, if you have a good binding pose, you can predict very well. If you don't have a good binding pose, you cannot predict. So this is a problem which is known in the field of docking forever. And people are working on solutions how to filter out the binding poses and make them better, if you wish. And I think that one of the ways we are working on is to using the information we've discovered from the same experiments retrospectively to filter them out. It's called a self-distillation process. So you can kind of distill the ground truth from your noisy data set. Like literally distill it out. Can we learn something about two receptors? Okay, we've done the prediction on one receptor, and that was quite a lot of work for a few master students and a postdoc kind of supervising them. And then we have to do some experimental validations, which was a few other PhD students were involved in it. And then we repeated the same process for another receptor, for beta-1 adrenergic receptor. It's very close. The binding sites are nearly identical. There's like 98% conservation. It's like 2 amino acids difference or 1 amino acid difference. And yet, there are ligands which are selective for one or the other. But what was most exciting when we did this analysis, we identified even the amino acids which are conserved, they're present in both receptors. In one case, they mediate agonism. In another case, they do nothing. So that's actually very interesting, because suddenly you can make an agonist on one receptor which would be not as good on the other receptor. And that's important because your beta-1 and beta-2 receptors, they're both present in the heart. And in beta-1 case, you want an antagonist to slow your heart down. In case of asthma, you want an agonist to open up your lungs. But the ligands are systemic, so they get everywhere. So you take your asthma medication, your heart starts beating. That's no good. And vice versa, you take your heart medication, then suddenly your lungs collapse. Also no good, right? So you really want to be selective. And the question, and we already started seeing these little differences, and that is quite amazing because it took a lot of years of research, and we've spent tens of postdocs years trying to figure out the answer to this question, and now we can calculate it with a lot less effort. So what's next? Well, I mentioned there are 350 GPCRs to go around. And for at least 50 of them, we have sufficient data in the public databases to do this type of calculations. And the question is, can we learn something which is common among all GPCRs? Is there kind of a universal recipe for their action? Interact with this residue, you always get an antagonism, and it seems like there is one. Because when you talk to medicinal chemists, you say, oh, I discovered if I interact with this residue, I have an antagonist. I say, oh, this one. So it looks like, because the architecture they're very similar, it looks like at least one or two interactions can be transferred from one to the other. So this is actually where we're going next. So with my collaborators in Birmingham, so we are applying for grants to repeat this exercise for every GPCR under the sun. And we even started collaborating with medics and pick a disease where we can test the hypothesis very quickly. So it's very interesting because it's a liver autoimmune disease, which doesn't really have a lot of treatment options. And we can identify targets and just verify it very quickly on liver tissues. Does it help with inflammation or not? And on this kind of explants, you don't have ethical concerns or no animals are being hurt in the process of your experiment, so that's easy. So this is just a reminder that we can use machine learning to uncover very detailed structure-activity relationship between the ligand properties and their signaling properties in GPCRs. And we can uncover the differences between otherwise very similar receptors. And I guess what's the next is to scale this up and to see how scalable is this process and what can be really useful in drug design. And on acknowledgement that there's always a lot more people involved than you think. I mean, it's very nice to give a talk and in 40 minutes tell you about fantastic work, but the reality is there's a lot of master students and PhD students and postdocs behind this. And the reality is that even 40 minutes talk, it probably means like 10 scientist years of work. And thank you very much for your attention. If you have questions.